welcome. So in this video, we will learn about the Kjeldahl method. Now, Kjeldahl method is considered to be as most accurate method to determine protein content in any of the food product, including the dairy products. Now, it is considered to be as most accurate method, and the Kjeldahl method consists of mainly three steps. First is digestion, second is distillation, and third is titration. So we will look into them one by one. So first, what happens is that firstly your sample is digested at a very high temperature in the presence of potassium sulfate using concentrated sulfuric acid, and also we are making use of catalyst as copper sulfate as catalyst, and we will see that all the elements which are present in the sample get converted to respective oxides, whereas your protein nitrogen is converted into ammonium sulfate by oxidation. So again repeating, the sample is digested using concentrated sulfuric acid at a very high temperature. Temperature goes to 410 to 430 degrees Celsius, and all the elements which are present in the sample get converted to their respective oxides, and your protein nitrogen is converted to ammonium sulfate by oxidation. So we will see in the coming slides about it in detail. Now, ammonium sulfate is reacted with strong alkali. Then the ammonium sulfate which is produced is then treated with strong alkali, and then we will see that ammonia will released when treating it with strong alkali, and ammonia will be released, and which by distillation is trapped in the boric acid. So this is the distillation process. So when the solution is made alkaline, your ammonia will be released with the help of steam. We are making use of steam, and then water also will be adding, and then by distillation the ammonia will be trapped in the boric acid containing the indicator. Okay, now once that ammonia is trapped in the boric acid, the solution is then titrated with a standard acid. We can use sulfuric acid, we can use hydrochloric acid, any of the acid we can use. Then finally we have to calculate the percentage nitrogen in the percentage protein in the sample from its weight. So we have how much weight we have the sample we have taken, and also how much volume the standard acid that has been used to titrate the ammonia. From that, these two values, we will be able to know how much is the protein content in the sample. So let us see it one by one. So first is the basic principle. What is principle? As I already told you, that in this sample is digested at a very high temperature, say greater than 420 degrees Celsius, using potassium sulfate and copper sulfate. So what will happen? Your all the elements, your carbon will be there, that will be converted to carbon dioxide. Your hydrogen will be there, that will be converted to water vapor. Your sulfur will be there, that will be converted to sulfur dioxide. Your phosphorus will be there, that will be converted to phosphorus pentoxide. Okay, and nitrogen which will be there, that will be converted to ammonium sulfate, as I have already told. Now, the potassium sulfate, purpose of using potassium sulfate in the mixture, in the solution, is to raise the boiling point of the reaction mixture during the digestion process, thereby increasing the effectiveness of the digestion process. So purpose of potassium sulfate is to raise the boiling point of the mixture in order to increase the effectiveness of the digestion process. Now, copper sulfate serve as the catalyst which fastens the digestion and also producing a clear reaction mixture. So copper sulfate enhances the speed of the reaction, and if apart from copper, we can also use selenium, mercury in the form of their salts can also be used, and they act as oxidation catalysts because they readily pass from a higher to lower valency, and vice versa also. So they are mostly catalysts which are generally used. So most of the time we are making use of copper sulfate only. Then digest is neutralized using an alkali, that is we are using sodium hydroxide, and thus liberating the ammonia. Ammonia will be liberated. The liberated ammonia is distilled off and trapped in boric acid. So boric acid will contain the mixer indicator that is titrated against standard acid. It may be HCl or H2SO4. Now, boric acid does not get involved in the titration but forms ammonium borate which reacts with ammonia. So we will see that boric acid is actually not involved in the reaction, but it first forms the ammonium borate, and then in the next step boric acid released as such. We will see that in the reactions. The formed ammonium borate complex is titrated with an acid, that is told HCl or H2SO4, using methyl red bromocresol green indicator. So this indicator is we are using. We can use any combination of indicator which can serve our purpose, which are fit to purpose. Methyl red, methyl blue can also be used. And then if equipment we are using that is from particular brand, they are also suggesting the use of mixed indicator as per their guidelines. We can use it. So till green distillation changes from colorless to pink. So when the ammonia will trapped in the boric acid, your color of the solution will change to green, and finally we have to titrate it using standard acids until we have the pink color endpoint. Okay, so some titer value will be there, and from weight of the sample taken we will be able to know percentage nitrogen, and from that we will be able to calculate the protein content in the sample. So let us see the reactions, because for any experiment reactions are very important part, so we must know the reaction. So as already told, first the solubilization stage, in which the sample containing protein is digested using concentrated sulfuric acid in the presence of sodium sulfate and potassium sulfate, or using selenium mercuric oxide or copper as catalyst, and we will have the ammonium sulfate formation. Your carbon will get converted to carbon dioxide, sulfur will get converted to sulfur dioxide, phosphor will get converted to phosphorus pentaoxide, water will be there, hydrogen will be there, then converted to water vapor. So elements are getting converted to their respective oxide, and your nitrogen will get converted to ammonium sulfate. And the next step involves the neutralization by alkali. So we'll add the alkali and then in the solution, and then we will see that ammonium oxide formation will be there, and on passing the steam your ammonium oxide will release the ammonia, and this ammonia will go to the boric acid. That is 50 ml of boric acid we are taking at the other end in the conical flask, to which the ammonia will go, and then we will have the color of the solution as green. Then last step is collection of ammonia and back titration. So ammonia is get trapped in the boric acid, that is ammonium borate formation will be there, and since pH increases to 8, so your color will be green at pH, because this indicator, combination of indicator at pH greater than 5.1 is having green color. And then this ammonium borate which is formed, that is titrated with HCl using and then we can see that the boric acid is released as such. So boric acid is actually not participating in the reaction. And then if H2SO4 will be used, then this will be the reaction which take will be the formation of ammonium sulfate, and if HCl is there will be formation of ammonium chloride. So as told that H2SO4 can also be used, and HCl also can be used, and these to be standardized before prior to use. Okay, now the volume of acid consumed during the titration is equivalent to the concentration of the ammonia, which is equivalent to the nitrogen content of the actual protein present in the sample. So this we can derive also in the coming slide. But here to say that the volume of acid which use the titer value will be the more is the titer value, this means that more of the concentration of more of the ammonia has been released, indicating that more of the nitrogen content is there in the protein sample. Now this gives the crude protein. The most important point is that the protein which we are getting is called as crude protein. Why it is crude? Because apart from nitrogen, some non-protein nitrogen also appears in forms the part of the nitrogen content. So apart from the protein nitrogen, we are having nitrogen coming from the non-protein nitrogen sources like urea, ammonia, creatinine. All these are the citrulline, all these are the, or some amino acids are there, all these are the NPN present in the milk. So nitrogen is coming from NPN also, so that's why it is not called as the true protein. It is not from Kjeldahl method we are not getting the true protein, but the crude protein. Okay, so again I can write here, right, it is crude protein. Okay, now this is the formula. So we have the percentage nitrogen content. So percentage nitrogen can come from this formula. So how this formula comes, let's see. Let us derive it this formula, because derivation is also N1 V1 equal to N2 V2. So here we are taking say here it is NaHCl, and here we are having the nitrogen. So we are treating the nitrogen with HCl. So your N1 will be equal to N2 V2. This is normality of HCl. Volume of titer that HCl used, that is titer value volume, and that is the this is the volume of the sample we have taken, or the weight of the sample we have taken. So this is because actually we have taken weight, so this can be changed to W. Okay, now normality of nitrogen. How will we write this? The weight of the nitrogen divided by because here a nitrogen element we are taking, so we will take atomic mass of the nitrogen into 1000 by into volume, that is 100 ml, because we are calculating percentage. So it will go atomic weight of nitrogen is 14.007. So yeah, it will be 10 So your WB by percentage in percentage, because we have taken per 100 ml, so it will be equal to 1.4007 into and normality of the HCl which we have taken into volume, the natural volume which you have obtained, and that is for sample, and we have to subtract blank also divided by mass of the sample taken, because we have taken the mass of the sample. So since the sample is taken in weight, so we will take here, we will place here W. ओके तो इस तरह से हम इस फार्मूले को ड्राइव कर सकते हैं. मैं एक बार दोबारा बता देता हूँ. N1V1 इक्वल टू N2V2. तो एक साइड हमें HCl लेना है, दूसरी साइड हमें जो है नाइट्रोजन होगा हमारा. तो V1 इधर आ जाएगा. V1 को हम W लिखते हैं क्योंकि हमने सैंपल जो है वेट में लिया है, तो हम W लिख लेंगे इसको. और हम अगर नॉर्मलिटी निकालें नाइट्रोजन, तो हमें पता है नंबर ऑफ ग्राम इक्विवेलेंट्स ऑफ सॉल्यूट प्रेजेंट इन 1 लीटर ऑफ सॉल्यूशन इज नथिंग बट द नॉर्मलिटी. अब इसको परसेंटेज निकालना है, तो हम 100 ml लेके यहाँ पर 10 लेके इसको 1.007 एटॉमिक मास होता है नाइट्रोजन का, और उससे हमें ये परसेंटेज नाइट्रोजन का ये फार्मूला आ जाएगा. तो हमें ब्लैंक भी लेना होता है, तो हम सैंपल में से जो सैंपल की जो टाइटर वैल्यू आएगी उसको ब्लैंक पे से हम टाइटर वैल्यू हम घटाएंगे. तो ब्लैंक की कैसे हमें करना है प्रोसीड, वो मैं आपको अभी बताऊंगा इसके नेक्स्ट स्लाइड. Okay, now whatever percentage nitrogen you are getting, that you have to multiply by 6.3 in case of dairy products. So dairy products के लिए हमें जो फैक्टर यूज करना पड़ता है, सॉरी डेयरी प्रोडक्ट्स के लिए जो हमें फैक्टर यूज़ करना पड़ता है वो 6.38 है. और ये आता कैसे है कि हमें पता है कि मिल्क प्रोटीन में ऐसा जो है जनरली जो है 15.65% नाइट्रोजन होता है. तो अगर हम 100 को 15.65 से डिवाइड कर देंगे तो हमारा जो आएगा वो 6.38 आएगा. इसलिए हम डेयरी प्रोडक्ट्स के लिए जो फैक्टर यूज़ करते हैं हम 6.38 करते हैं, क्योंकि नाइट्रोजन कंटेंट जो है डिफरेंट प्रोटीन्स में, डिफरेंट फूड प्रोडक्ट्स में डिफरेंट होता है. तो ये जो फैक्टर है वो कन्वर्जन फैक्टर जिसे हम बोलते हैं CF, वो हर एक सैंपल के लिए अलग अलग होता है. और इनकी क्या क्या वैल्यू है वो हम अभी कमिंग स्लाइड में देखेंगे. लेकिन डेयरी प्रोडक्ट्स के लिए जो फैक्टर यूज़ होता है वो 6.38 होता है, कंसिडरिंग 15.65% nitrogen in the milk proteins. Okay, now proteins in general vary from 14 to 90%. So different products it is having 14 to 90%, it varies, and thus a universal conversion factor cannot be used for them. So different type of food product we are making use of different conversion factor. For example, egg or meat we are making use of 6.25, dairy products it is 6.38, for wheat we have to use 5.7, other cereal grains and oilseed we have to use 6.25, almonds we have to make use of 5.18, peanuts and Brazil nuts conversion factor is 5.46, other tree nuts and coconut 5.30. So depending on the protein content in the sample, different conversion factors are there for different food products. Okay, now let us start the procedure. So first we have to know what are chemicals or what are reagents which are being used. So let us see. So we will discuss the method by IDF method. We will now discuss now of what are the chemicals which are used. First, potassium sulfate, that is raised to raise the boiling point of the mixture. That should be nitrogen free or low in nitrogen content, because ultimately we have to determine the nitrogen content in the sample. And also then we have to use copper sulfate solution, which will be acting as catalyst. So we will make 5% solution by dissolving 5 gram of copper sulfate pentahydrate and making up the volume to 100 ml in a 100 ml volumetric flask. So we will have the 5% copper sulfate solution. Then concentrated sulfuric acid will be used to digest the sample, and sodium hydroxide to be prepared 50% mass by mass. That meaning that if you dissolve 50 gram of sodium hydroxide pellets in water and finally make up the volume to 100 ml. So this is mass by volume actually. Sorry, this is mass by volume. So 50 gram sodium hydroxide, you take dissolve 50 gram sodium hydroxide pellets in water and make up the volume to 100 ml. You will have the 50% mass by volume solution of the sodium hydroxide. Now boric acid, you have to prepare 4% boric acid solution you can prepare. So you take 40 gram of the boric acid in hot water and then allow the solution to cool and then make up the volume to 1 liter. And then you can add the 3 ml of the indicator which we will be preparing in the below line. So as per the below line, so 3 ml of the methyl with bromocresol indicator solution you can add, and then store it the solution in borosilicate glass bottle for further carrying out the experiments. Now indicator solution how to be prepared, that you take 0.1 gram of the methyl in 95% ethanol, dilute to 50 ml with ethanol. So it is to be prepared in 95% ethanol only. Then also you dissolve 0.5 gram of bromocresol group in 95% ethanol and make up the volume to 250 ml with ethanol. And then you can mix 1 part of the methyl solution and 5 parts of the bromocresol group solution simply you combine both the solution, you will have the indicator solution. So as already told, instead of methyl red and bromocresol green, you can also use methyl red and methylene blue. But for the as per the instruction of the manufacturer or the instruction or the guidelines of the equipment provider, we can use the indicator. So that simply the indicator have to serve a purpose that is fit for use here. So we are making use of methyl red solution and bromocresol green solution. Okay, now we have to use the hydrochloric acid also, that will be 0.1 normal, that is used for titration. Ammonium sulfate should be to be used. The purpose of ammonium sulfate is to check the accuracy of the procedure, and then tryptophan or lysine is to check the accuracy of the digestion, that is minimum 99% acid to be used. This for accuracy of procedure, this is for accuracy of digestion. And then sucrose is used for the carrying out the blank test and should not have nitrogen more than 0.002%, that it is used for carrying out the blank test. Okay, now see the step-by-step procedure how to perform the experiment. So add to a clean and dry Kjeldahl flask. So Kjeldahl flasks are available. You add 5 to 10 boiling beads you can add, and then you have to add 12 grams of the potassium sulfate, 1 mol of the copper sulfate solution, and the quantity of the sample is to be decided depending on the protein content of the sample. So how to it is to be decided that the sample should contain 0.2 gram, 0.2 to 0.4 gram of the nitrogen. In this way, it has to be decided how much weight of the sample we take on. Say a product is having, say, 25%, we have a protein content is there in a sample. We know that a sample is having 25% protein content. So obviously in 100 gram there will be 25 gram, in 10 gram there will be 2.5 gram, and in 1 gram it will be 0.25 gram nitrogen, so which is falling between this range. So I can use 1 gram of the sample for the digestion instead of taking 5 gram. Okay, so this way the quantity of the sample to be taken depends on the protein content in the sample. Okay, and it is to be decided in a way that the sample should have the nitrogen content between 0.2 to 0.4 gram, as I have told you. So and then you have to add 20 ml of the concentrated sulfuric acid. Now use the 20 ml acid to wash down any copper sulfate solution. So that will be, so if a solution is there adhering to the glass of the tube, that to wash down we can use the acid, and also potassium sulfate or milk left on the neck of the flask. So any sample is that is adhering to the glass, that to wash down the same, the acid can also be used. Then gently mix the content of the sample. You have to mix the content of the sample, and then you have to go for the digestion. So this is you can say that this is ND force Kjeldahl machine. Force ND machine is there, and then here we can see that there are 8 samples can be digested at a time, and then here you will be placing all the tubes at the positions, and then we will cover this cover will place on the tubes, and then you have to set the digestion block at initially you have to carry out at initial temperature so as to control foaming. You will initially keep the temperature at 180 to 130 degrees Celsius for 30 minutes. You will allow it to digest at lower temperature so as to control the foaming, and then transfer the tube to the digestion block and place the exhaust manifold, which is itself connected to water jet with the suction rate of the water. Now there is a fume control mechanism is there. A mechanism may be of different. Some manufacturer having scrubbers, and some may directly use the tap pressure. So suction will be created by the running water from the tap, and then fumes will be carried away by the water in the drain. So that way also we can you can see the video also in this channel. We will be able to know how the things are working. Now you digest the sample until white fumes develop, and then increase the temperature of the digestion. So then you have to increase the temperature to 410 to 430 degrees Celsius and continue to the digestion till the digestion is clear. So until the digestion is clear, you have to continue the digestion. Generally it is completed within 2 hours or less than that also. Some of the samples, and the rest portion should be digested 15 minutes. Once it is clear, then you can 15 minutes more time you can take, and after the solution has been cleared. Now remove the tube from the block with the exhaust manifold in place, and then allow the tubes to cool for 15 minutes. Once the tubes are sufficiently cool to handle, remove the exhaust manifold. Okay, then comes the distillation. So this is again post distillation machine. Here we can set the program, and then we know that once that we can place tube here to the digestive sample in the tube to be placed here. So this is the position, and here we will keep the conical flask containing 50 ml of the boric acid. So which will boric acid containing the indicator which will trap the ammonia coming after the distillation. So this is the program we have set. Say you have to take about 70 so one by one automatically first the water will come, that is 70 ml of the water will come, and then 80 ml of the alkali will come to release the ammonia, and the distill time is kept at here 4 minutes, and then steam is 80% to which will facilitate the release of ammonia from the sample. That is once the ammonium sulfate is formed, and from the ammonium hydroxide the ammonia will be released by the using 80% steam. Now transfer the digestion tube to the distillation, and then the program for automatic distillation. So one by one water will come 70 ml, then alkali 80 ml will come, then distillation is kept at 4 minutes. The steam used to be used at 80% efficiency capacity. Now place a conical flask containing 50 ml of boric acid solution in the outlet of the condenser in such a way that the delivery tube is below the surface of the excess boric acid solution. So here we will keep the conical flask, then adjust the distillation unit to dispense outlet to 80 ml, and which will come 70 ml of water will come distill the ammonia liberated by the ammonia and collect the distillation in the excess boric acid. So you will see that once the distillation is completed after 4 minutes, around 150 ml of the solution will be collected in the conical flask, and the color will change to green because ammonia is entrapped, and indicator will change the color to green because the pH will increase to around 8. Then the final step of the Kjeldahl method is titration. So we titrate the boric acid receiving solution with standard HCl solution to the first trace of the pink color. So as soon as pink color will come, then during the titration, that will be your titer value. Take the burette reading to at least the nearest 0.05 ml, and the latest plate can also be used to visualize the endpoint that is there. Now we have to always perform the blank to overcome any of the effects because of the solvent tubes and all the materials which are being used in the process to if any contamination is there. To overcome all those effects, we are making use of blank, and then you have to carry out a blank test following the procedure described above. Taking instead of 5 ml of milk, you will take 5 ml of water, and then you have to take 0.85 gram of sucrose instead of the test procedure. For blank carrying of the blank test, you have to take 0.85 gram of the sucrose, because sucrose is considered free of nitrogen, and then if some quantity is there, then that has to be subtracted. Then always when we are performing the Kjeldahl method estimation, that is we are determining the protein by Kjeldahl method, we have to carry out the nitrogen recovery test. So in these two major things to be done. One is we have to check the accuracy of the procedure. That should be in accordance as with the procedure as follows. So no loss of nitrogen occurs by using a test procedure. So we are making use of accuracy. For checking the accuracy of procedure, we are making use of 0.12 gram of ammonium sulfate along with a 0.85 gram of the sucrose, and then all the reagents, all the procedure which has been discussed to be followed here also. You digest and distill under the same condition as for the milk simulation. So here the accuracy of the procedure is determined by calculating the nitrogen content in the ammonium sulfate, and approximately nitrogen content in ammonium sulfate is around 21.9%, and then again you can verify using that is there on the label which ammonium sulfate you are using. Then the percentage of nitrogen recovered should be between 99 to 100% for the given apparatus, and if it is there, then we say that the procedure is accurate. And if once it is declared that the procedure is accurate, you have to also check the efficiency of digestion also, and that is being done either using lysine hydrochloride, 0.16 gram of the lysine hydrochloride, which contain around 15.33% nitrogen, or you can use 5.18 gram of tryptophan, which contains 30.72% nitrogen, along with 0.62 gram of the sucrose. So all the conditions, all the digestion and distillation again to be done as has been discussed for the milk. At all the reagents as a milk sample, digest and distill under the same condition as for the milk sample, at least 98% of nitrogen shall be recovered. Okay, so it's the recovery should not be less than 93 98%, and if it is less than 98% after having a 99 to 100% recovering on ammonium sulfate, then the temperature or the time of the digestion is insufficient. That there is, this may be, if it is less than 98%, then there may be two reasons for that. One is the temperature or time of digestion is insufficient, and also there is undigested sample material which is there on the inside of the Kjeldahl pack. So there is undigested sample also maybe there, because of that also the recovery is coming less than 98%. Now again, this way you have to check the accuracy of procedure, also accuracy of the digestion also regularly. Now this formula I have also told you that how to derive this formula I have already told you. So this way you will be calculating the percentage by mass of the nitrogen, and then you have to multiply by 6.38, which is the factor which for dairy products, and then I have told you that these are the conversion factor to be used for different products. And then if you see that depending on the protein content of the sample, how much sample of how much weight of the sample to be taken. So here the percentage protein content in different foods are given, like apple raw with skin, that 0.2 gram protein is only there, 0.2% is there. In egg raw whole, 12.5% protein is there, and then ham sliced and regular, that is 7.6% protein is there. And depending on the protein content in the sample, the weight of the sample to be taken for determination of protein content will be decided. So here it is written also that the protein content of different types of milk and milk products varies. So for determination of total protein content by Kjeldahl method, the size of the sample varies, so that the test portion of the sample should contain 0.2 to 0.4 gram of the protein, that has already told you. So these are the protein content in different milk products. So depending on the protein content, if we know, and then accordingly we can take that amount of samples. So for example, say any of the sample you take. Here also some other products are given. Cheddar cheese is there in fat food. For example, let's say in fat food is having 22% nitrogen. So 1 gram will contain 100 gram will contain 10 will 2.22. Again, this is falling in the range of 0.2 to 0.4. So I can take 1 gram of the sample of infant food for the digestion purpose for determining the protein content. So that way we can decide the weight of the product to be taken to calculate to determine the protein content in any of the sample. Now, why to go for Kjeldahl method? Why to determine the protein content in different samples? So one is that nutrition labeling. Say if any new product is being manufactured, new product has been developed by NPD, and we want to label how much protein, how much fat, how much different level constituents are there. Then for protein determination, we can go for nutrition labeling. Functional property. So there are so many functional property of the proteins are there, like foaming, emulsification, gelation, viscosity, water absorption capacity, solubility. So many functional properties are there that obviously will depend on the different types of protein in the sample, and then overall protein will be required for its determination. Now, proteins of food have unique functional properties that I have already told you. Different functional properties I have discussed, like gliadin and gluten in wheat flour for for providing elasticity. These protein having their the role. So we have to define sometimes individually proteins also. Then for biological activity determination, including enzymes and enzyme inhibitors. Enzyme activity is expressed as terms of specific activity, the unit of enzyme per milligram of the protein. So specific activity can also be determined. So all these things will depend on the quantity of the protein which is there in the sample. Now, protein analysis is required if you want to know the total protein content of the particular protein in nature, and that is obvious that if you want to determine casein, you isolate casein. You want to determine any other protein, whey protein, you isolate whey protein, then determine using Kjeldahl method protein content during the isolation and purification of protein. So during different steps are there for isolating in protein. At each step you can determine the protein content using Kjeldahl method, and then you can calculate. Obviously you treat the sample of milk with 12% EC, and then you will see that all the protein will get precipitated, and you determine the NPN in the filtrate. Amino acid composition can be determined. Net value, that is the protein efficiency ratio, biological value can be determined. Told that the activity of the enzyme can also be determined. So this is the purpose why we go for the determination of the protein content in different samples. Okay, now we will see the practical part of it. I will run the practical also now. Okay, now we will see the practical of Kjeldahl method.
वेलकम वेलकम वेलकम। तो आज हम जानेंगे कि हम Kjeldahl मेथड से किसी भी मिल्क प्रोडक्ट्स में कैसे हम प्रोटीन कंटेंट निकालते हैं। तो ये पर्पस देख सकते हैं, डाइजेशन ट्यूब है, इसमें पहले हम सैंपल डालेंगे। देखिए हमने 0.5 ग्राम पाउडर सैंपल लिया हुआ है, ये हमने जल्दाल ये डाइजेशन ट्यूब में डाल दिया है। इसके बाद हमें 12 ग्राम जो है पोटेशियम सल्फेट डालना होगा। इसका पर्पज़ होता है मिक्सर का बॉइलिंग पॉइंट बढ़ाने का। इसके बाद हमें इसमें 1 ml जो है कॉपर सल्फेट सोल्यूशन डालना है। डालने के बाद हमें इसमें 20 ml जो है कंसनट्रेटेड सल्फ्यूरिक एसिड डालना होगा, जो कि सैंपल को डाइजेस्ट करेगा। ये देखिए हमने डाल दिया है। सैंपल है, हम उसको डाइजेस्ट करेंगे। डाइजेशन के लिए जैसे मैंने आपको बताया था, ये हमारे पास जनरल ट्यूब है। इस ट्यूब के अंदर हमने 0.5 ग्राम जो है पाउडर सैंपल लिया हुआ है। इसके अलावा इसमें हमने कॉपर सल्फेट सोल्यूशन 1 ml डाला हुआ है और पोटेशियम सल्फेट 12 ग्राम डाला हुआ है, और हमने 20 ग्राम जो है 20 ml जो है कंसनट्रेटेड सल्फ्यूरिक एसिड भी डाला हुआ है, ताकि जो हमारा ऑर्गेनिक नाइट्रोजन है उसको हम अमोनियम सल्फेट में कन्वर्ट कर सकें और जो हमारा बाकी ऑर्गेनिक मैटर है वो रिमूव हो जाए। इन डाइजेशन ट्यूब्स को हम जो पोजीशंस बनी हुई है इस हमारे डाइजेशन ब्लॉक में, तो हम इन पोजीशंस पे रखेंगे। और हमें जो टेम्परेचर सेट करना होता है इनिशियली, वो हमने इनिशियली जो हम इसमें टेम्परेचर सेट करेंगे वो 220 डिग्री सेल्सियस पे करेंगे, क्योंकि हमें 220 डिग्री सेल्सियस पे 30 मिनट के लिए रखना होगा। ताकि हम इनिशियली लो टेम्परेचर इसलिए रखते हैं ताकि जो कोई भी फोम फॉर्मेशन है हम उसको कंट्रोल कर सकें। तो चलिए हम देखते हैं कैसे हम असेंबली को प्लेस करते हैं। देखिए ये हमने डाइजेशन ट्यूब को प्लेस करना है। ये हमारे पास देखो डाइजेशन ट्यूब्स हैं, इसमें हमने इसको प्लेस कर दिया। तो इसके ऊपर हम इसका असेंबली को कवर कर देंगे। तो इधर आप देखिए हमने पहले टाइमिंग सेट करते हैं यहाँ पे, ये देखिए। तो देखिए हमने 220 डिग्री सेल्सियस 30 मिनट के लिए हमने यहाँ पे सेटिंग कर दिया है। तो इनिशियली हम लो टेम्परेचर पे क्यों ला रहे हैं, क्योंकि हमें फॉर्म को कंट्रोल करना दिखाना है। स्टार्ट नहीं किया, तो देखिए हम स्टार्ट का बटन दबाएंगे। देखिए हमने स्टार्ट का बटन दबा दिया। देखिए जैसे हमारी फ्यूम्स बनेंगी तो फ्यूम्स का रिमूव करने का मैकेनिज्म भी है हमारे पास। ये देख सकते हैं हमारे पास ब्लैक ट्यूब है, तो इससे सारी जो फ्यूम्स जो बनेंगी वो हम वो हमारी ड्रेन में जाएंगी। देख सकते हैं पानी के थ्रू जो है ड्रेन में जाएगी, क्योंकि यहाँ पे कम प्रेशर है और वहाँ पे प्रेशर हमारा ज़्यादा है, तो सक्शन मैकेनिज्म से हमारे जो फ्यूम्स है वो ड्रेन के अंदर जाएंगे। तो जैसे मैंने आपको बता रहा था कि ये हमारे पास डाइजेशन यूनिट है। डाइजेशन का क्या पर्पज होता है कि मैंने आपको बताया कि सैंपल इज डाइजेस्टेड यूजिंग कंसनट्रेटेड सल्फ्यूरिक एसिड इन द प्रेजेंस ऑफ पोटेशियम सल्फेट एंड कॉपर सल्फेट and at a very high temperature. Temperature goes to 410 to 430 degrees Celsius. So in this way, what is happening is that we are digesting the sample. So whatever carbon is there, that will be converted to carbon dioxide. Hydrogen will be converted to water vapor. Your sulfur will get converted to sulfur oxide. Your phosphorus will be converted to phosphorus oxide. So all the other elements will get converted to their respective oxides, and your nitrogen will get converted to ammonium sulfate. So during digestion, your nitrogen which is present in the sample is getting converted to the ammonium sulfate. तो इसमें जैसे मैंने बताया आपको, कॉपर सल्फेट का जो पर्पस होता है वो कैटलिस्ट का काम करता है। तो रिएक्शन को तेजी से करने के लिए हम कॉपर सल्फेट का यूज़ करते हैं। पोटेशियम सल्फेट जो हम इसमें डालते हैं वो बॉइलिंग पॉइंट, जाके मिक्सर का जो बॉइलिंग पॉइंट है वो बढ़ाया जा सकते हैं, इसलिए हम पोटेशियम सल्फेट इसमें डालते हैं। तो देखिए हमारा आधा घंटा कंप्लीट हो चुका है। 220 डिग्री सेल्सियस पे हमने आधे घंटे के लिए सैंपल को डाइजेस्ट कर दिया। इसके बाद हमें इसका टेम्परेचर बढ़ाएंगे। तो देखिए हमें 2 घंटे का टाइम देना है। हाँ जी, और टेम्परेचर जो है हम 420 रखेंगे। हाँ जी, देखिए अब स्टार्ट का बटन दबाएंगे तो हम देखेंगे कि टेम्परेचर जो हमारा 420 पे आएगा, और इस टेम्परेचर पे हमें सैंपल को डाइजेस्ट करने देना होगा 2 घंटे के लिए। तो देखिए हमारा डाइजेशन कंप्लीट हो चुका है, और हम देख सकते हैं कि जो हमारा जो सैंपल थे देख सकते हैं वो बिल्कुल क्लियर हो चुके हैं। तो ये इंडिकेट कर रहा है कि हमारा जो डाइजेशन है वो कंप्लीट हो चुका है। तो चलिए अब देखते हैं हम जो हमारे डाइजेस्टिव सैंपल हैं उनका हम डिस्टिलेशन कैसे करते हैं। तो आप ये देख सकते हैं हमारे पास जो है जलदाल की फोस की जो है जलदाल की यूनिट है, ये डिस्टिलेशन यूनिट है फोस की। और ये आप 4 कंटेनर देख सकते हैं नीचे। हमारे पास पहला जो कंटेनर है वो वाटर का है, इसके अंदर हम जो पानी डिस्टिलेशन के लिए जो यूज़ होगा वो हम इसमें हमने बोरिक एसिड लेते हैं, अदरवाइज हम इसको मैनुअली भी डाल सकते हैं कोनिकल फ्लास्क में। इसके बाद ये ड्रेन वेस्ट होती है, जो भी हमारा वेस्ट जब हम वॉशिंग करते हैं अपने ट्यूब्स की या पूरे अपरेटस की, तो जो वास्ट पानी होता है वो वॉशिंग के बाद जो है पानी वो यहाँ पे कलेक्ट होता है। ये देखिए हमारी ये पोस की जल्दा की मशीन है, और इसमें अगर हमें प्रोग्राम हमने सेट कर सकते हैं, आप देख सकते हैं यहाँ पे हमने प्रोग्राम सेट किए हुए हैं। हम ये देखिए आप इसको ये प्रोग्राम सेट हुए हुए हैं, और इसमें आप कोई भी प्रोग्राम ले सकते हैं। जैसे हम जो है 6th लेंगे, 6th प्रोग्राम हमने बनाया हुआ है जो हमारे सैंपल के लिए, और देख सकते हैं इसमें हमने पानी जो है 70 ml जो हमारा डाइजेस्टेड ट्यूब जो सैंपल है ट्यूब के अंदर, वो हम यहाँ पे रखेंगे। यहाँ पे जो इसकी पोजीशन है अभी आपको हम करके भी दिखाएंगे। जो डाइजेस्टेड ट्यूब है, डाइजेस्टेड जो ट्यूब है हमारी डाइजेशन ट्यूब, वो हम यहाँ पे रखेंगे। जो हमारा बोरिक एसिड है, ये देखिए हमने बोरिक एसिड यहाँ पे कलेक्ट कर रखा है। इसके अंदर हमने इंडिकेटर डाला हुआ है आपका मिथाइल रेड, और प्रोमोक्सोल ग्रीन जो है इंडिकेटर हमने इसमें मिक्स कर रखा है। दोनों का 0.2% सोल्यूशन हमें 95% इथेनॉल में मिलाना है, और एक पार्ट जो है मिथाइल रेड का, और 5 पार्ट्स जो है ब्रोमोक्रिसोल ग्रीन का हमें मिक्स कर देना है, तो हमें ये मिक्स इंडिकेटर मिल जाएगा। ये जो मिक्स इंडिकेटर है वो हम यहाँ पे रखेंगे, या पोजीशन इसकी ये है। और जब रखने के बाद आप देख सकते हैं यहाँ पे अगर प्रोग्राम देखें तो इसमें हमें 70 ml हमें पानी आता है बारी बारी से। पहले 70 ml जो पानी आएगा, और पानी क्यों चाहिए, तो डिस्टिलेशन होना है। अल्कली 80 ml हमें चाहिए, जो 50% हमने एनुअल सोल्यूशन अभी मैंने आपको फ्लास्क कंटेनर में दिखाया था, तो वो जो है उसका पर्पज़ है अमोनिया को रिलीज़ करना। उसके बाद हमारा डिस्टिलेशन टाइम जो है 4 मिनट का सेट किया हुआ है, तो 4 मिनट में हमारा लगभग 150 ml जो है डिस्टिलेट कलेक्ट हो जाता है। उसके बाद हमारे स्टीम जो 80% पे हमने रखी हुई है, तो स्टीम जो है पर्पज़ यही है कि आपका जो अमोनियम सल्फेट है उससे अमोनिया रिलीज कराने में आपको स्टीम जो है हेल्प करेगा 80%। तो चलिए अब हम देखते हैं कि स्टेप बाय स्टेप हम डिस्टिलेशन प्रोसेस कैसे करते हैं। देखिए हमारे पास ये डाइजेशन ट्यूब है, इस ट्यूब को हम अपनी पोजीशन पे प्लेस करेंगे। देखिए हम जो डाइजेस्टिव जो ट्यूब हमारे पास है, जिसमें हमारा डाइजेस्टेड सैंपल है, उसको हमने प्लेस कर दिया। और ये जो दूसरी फ्लास्क है, इसमें हमने 30 ml जो है बोरिक एसिड कंटेनिंग इंडिकेटर मिक्स इंडिकेटर हमने डाला हुआ है, और ये अपनी पोजीशन पे हम यहाँ पे रख देंगे। इसके अलावा मैंने आपको अभी प्रोग्राम भी दिखाया था, तो ये हम प्रोग्राम दोबारा देख सकते हैं। यहाँ पे हमने पानी जो है 70 ml आएगा, इसलिए 80 ml आएगा, और डिस्टिलेशन टाइम हमने 4 मिनट रखा हुआ है। तो अब हम सिर्फ हमें इतना करना है कि हमें स्टार्ट का बटन दबा देना है। तो देखिए ये अपने आप क्लोज हो रहा है, आप देख सकते हैं असेंबली क्लोज हो चुकी है। और अभी जो डिस्टिलेशन है हमारा प्रोसेस है, ये स्टार्ट होगा। तो जैसा मैंने आपको बताया था, पहले पानी आएगा, और उसके बाद हमारा NaOH आना चाहिए। NaOH जो है हमने 80 ml दिया हुआ है, तो देखिए ये हम NaOH डाल रहे हैं, 80 ml NaOH आ चुका है। और ये देखिए स्टीम भी आना शुरू हो गई है, देख सकते हैं। तो स्टीम से जो है डिस्टिलेशन होता है, तो अमोनिया रिलीज हो रही है, और अमोनिया जो है आप देखते हैं डिस्टिलेशन होते हुए इस कोनिकल फ्लास्क में आना शुरू हो गया है, देख सकते हैं आप। तो अब ये डिस्टिलेशन हम 4 मिनट तक होने देंगे, और हम एक्सपेक्ट कर रहे हैं कि हमें 150 ml डिस्टिलेट जो है हमारा कलेक्ट हो जाएगा आफ्टर 4 मिनट। तो देखिए हमारा ग्रीन कलर आना शुरू हो गया है, देख सकते हैं आप कोनिकल फ्लास्क में। इसका मतलब क्या है कि आपका अमोनिया जो है सैंपल से आपकी फ्लास्क में ट्रांसफर हो रही है। देखिए ग्रीन कलर हो गया है। अब ये डिस्टिलेशन प्रोसेस जैसा कि हमने कमांड दी हुई है, ये 4 मिनट तक रेगुलर चलता रहेगा। तो देखिए हमारे, हाँ जी देखिए, तो हमारे 4 मिनट होने वाले हैं। हाँ जी देखिए हमारा डिस्टिलेशन कंप्लीट हो गया है। तो देखिए अब इसका जो कवर है वो ऑटोमेटिकली रिमूव होगा। तो देखिए हमारा कवर रिमूव हो चुका है। अब हम तो देखिए हम जो कोनिकल फ्लास्क हम उठाकर अब इसका टाइट्रेशन करेंगे, जो लास्ट स्टेप होता है टाइट्रेशन का। तो चलिए अब देखते हैं जो हमारा डिस्टिलेट कलेक्ट हुआ है, इसका हम टाइट्रेशन करेंगे। तो जैसे मैंने आपको बताया कि जेल्डाल मेथड में 3 स्टेप्स होते हैं, पहला डाइजेशन होता है, दूसरा डिस्टिलेशन होता है, तीसरा टाइट्रेशन होता है। तो जैसे हम देख सकते हैं ये जो डिस्टिलेट हमारा कलेक्ट हुआ है ग्रीन कलर का है, क्योंकि इसके अंदर अमोनिया ट्रैप्ड है। और ये हमने टाइट्रेशन कर रहे हैं, हम 0.1 नॉर्मल HCl से हम टाइट्रेशन कर रहे हैं। तो हम एंड पॉइंट जो एक्सपेक्ट कर रहे हैं, जो हमारा एंड पॉइंट आएगा वो आपका पिंक कलर आएगा, और जो हमारी टाइटर वैल्यू होगी उसको हमें फार्मूले में पुट करना है। दैट इज फार्मूला इज 1.4007 इंटू वॉल्यूम यूज्ड फॉर द सैंपल माइनस वॉल्यूम यूज्ड फॉर ब्लैंक इनटू नॉर्मलिटी ऑफ HCl डिवाइडेड बाय वेट ऑफ द सैंपल टेकन। तो हमने वेट सैंपल 0.5 ग्राम वो लिया हुआ है ये पुट करके, और जो भी हमारी नाइट्रोजन वैल्यू आएगी परसेंटेज नाइट्रोजन, उसको हम 6.38 से मल्टीप्लाई कर देंगे तो हमारा क्रूड प्रोटीन परसेंटेज आ जाएगा। देखिए हमारी टाइट्रेशन अभी कंटिन्यू है और ये देखिए कलर चेंज होता हुआ हमें नज़र आ रहा है। देखिए थोड़ा स्लाइड चेंज हो चुका है, अभी ज़्यादा देर नहीं होगी, ये देखिए। हाँ जी बस, तो ये हमारा जो कलर हम देख सकते हैं, ये पिंक कलर हमारा आ गया है। और हम अगर वैल्यू नोट करें यहाँ पे, हमारी जो टाइटर वैल्यू आ रही है वो आपकी 8 ml आ रही है। तो चलिए अब हम कैलकुलेशंस देख लेते हैं। तो जैसे कि आप देख सकते हैं यहाँ पे परसेंटेज नाइट्रोजन जो है उसका फार्मूला दिया हुआ है 1.4007 इंटू वी एस माइनस वी बी डिवाइड इंटू नॉर्मेलिटी डिवाइड बाई डब्ल्यू। तो वी एस जो है आपका वॉल्यूम ऑफ HCl यूज्ड फॉर सैंपल है, वी बी जो है वॉल्यूम ऑफ HCl यूज्ड फॉर ब्लैंक है, एन जो है नॉर्मेलिटी ऑफ HCl यूज्ड है, डब्ल्यू इक्वल टू वेट ऑफ सैंपल टेकन है। और इसी तरह से अगर प्रोटीन परसेंटेज निकालें जो WP है आपका परसेंटेज बाय मास, तो जो भी आपकी परसेंटेज नाइट्रोजन आएगी उसको आप अगर 6.38 से मल्टीप्लाई कर देंगे तो आपका क्रूड प्रोटीन परसेंटेज जो आपके सैंपल में वो निकल आएगा। तो 6.38 कहाँ से आता है कि अगर हम जानते हैं कि जो दूध के अंदर जो प्रोटीन होता है उसके अंदर जो नाइट्रोजन होती है वो अप्रोक्सीमेटली 15.65% होती है। तो अगर 100 को आप 15.65 से डिवाइड कर देंगे तो आपको 6.38 मिल जाएगा। तो ये फैक्टर वहाँ से आता है। और दूसरा ये जो 1.4007 है ये एक्चुअली एटॉमिक वेट ऑफ नाइट्रोजन से आता है, तो हम डिराइव भी कर सकते हैं इस फार्मूले को। तो देखिए जो हमारी वैल्यूज़ आई हैं वो हम अगर पुट कर देते हैं, देखिए हमारी ब्लैंक के लिए जो वॉल्यूम टाइटर वॉल्यूम आया था वो 0.2 ml आया था, और जो हमारे सैंपल के लिए अभी हमने देखा 8 ml आया है। और अगर हम सारी वैल्यूज़ जैसे नॉर्मल एन की वैल्यू 0.1 नॉर्मल रख के, डब्ल्यू की वैल्यू 0.05001 ग्राम रख के अगर हम पुट कर देते हैं तो हमारा नाइट्रोजन परसेंटेज जो आ रहा है वो 2.18% आ रहा है। और इसी को अगर हम 6.38 से मल्टीप्लाई कर दें तो हमारा प्रोटीन कंटेंट निकल आएगा जो कि 13.91% है। तो इस तरह से हम प्रोटीन कंटेंट किसी भी मिल्क एंड प्रोडक्ट में जेलरल मेथड से डिटरमाइन करते हैं। धन्यवाद।